Hello everyone, I am Assistant Professor V Shete from Department of Microbiology, KG Somaya College. In this online lecture, we will study the effect of antimicrobial agent on the bacteria. In that, we will study the enzyme inhibitors, the molecules or the antimicrobial agents which are responsible for inhibiting the enzymes which are very essential or very important in carrying out various processes. These are called as the enzyme inhibitors. In the last online lecture, we have studied the protein synthesis inhibitors and the nucleic acid inhibitors. In the enzyme inhibitor, the first is the trimethoprim. Okay, we will study the trimethoprim. It is the enzyme. It is the antimicrobial agent and antibiotic which is responsible for synthesis inhibition of enzyme production in the bacterial cells. Now, the trimethoprim is the bacteriostatic antibiotic. Bacteriostatic, which will inhibit the bacteria, which will not kill the bacteria. Okay, static is for inhibition. Then trimethoprim is the diaminopyrimidine related pyrimethamine. Okay, then which is selectively inhibit the bacterial DHFRase, which is called as the dihydrofolate reductase enzyme. Okay, now this enzyme is responsible for producing the folic acid. Okay, with the help of the pteridine and the PABA, that is the PABA, it will produce the folic acid along with the glutamic acid. Alright, then trimethoprim is more than 50,000 times active against the bacterial DHFRase against the mammalian enzyme. It is mostly used for treating the urinary tract infections as well as the respiratory tract infection. Okay, now what is the trimethoprim is doing basically? Trimethoprim will inhibit the production of the folic acid by inhibiting that dihydrofolate reductase enzyme. Okay, now the dihydropteroid acid synthesis from pteridine plus PABA, that is the para-aminobenzoic acid, you can see in that picture. Okay, then this will convert and this will produce the pteridine and the PABA complex. Now this complex will attach to the glutamic acid, alright, and these three together will be responsible for producing the folic acid. Okay, now this trimethoprim, this is the normal reaction that you can see in the above one. The first one is the normal reaction. Okay, now in the presence of the trimethoprim is the structural analog of that pteridine. Okay, so what it will do, it will fold the bacteria. Okay, because it is only structurally similar, not chemically similar. Instead of producing the folic acid, it will inhibit the folic acid production. Now as it is a structural analog of the pteridine, it will bind to the PABA. Okay, here it is mentioned as the sulfa drug, which is also has the similar action. Okay, So here is the structural analog of the trimethoprim, the red color one, and therefore you can see there is mentioned the fold. First enzyme is fold, okay, and then there will be the addition of the glutamic acid, and now this 3 will not able to produce the folic acid. Why? Because now in this case there is not the pteridine is present, there is the trimethoprim is present. Okay, so therefore this is how it will inhibit the enzyme process. Okay, therefore it will inhibit the second enzyme, which is the folic acid. Okay, then trimethoprim can be given. This is the trimethoprim reactions in the normal and in the presence of trimethoprim antibiotic drug. Now we will see what is its spectrum: broad spectrum, narrow spectrum. Okay, so the trimethoprim is the bacteriostatic antibiotic. It is active against gram negative as well as the few gram positive. So it is coming down under the gram wide spectrum, broad spectrum antibiotic. Okay, then the sulfonamide and the trimethoprim both are bacteriostatic. Okay, in some cases if we administrate both the drugs together, then that can result into the bactericidal results in some of the bacteria. Now what are the pharmacokinetics? By means how we can take this drug. So we can take this drug orally, which is fully absorbed in the GI tract and distributed along throughout the tissue and the body fluid. Then it reaches the high concentration in lungs and the kidneys. Okay, then the trimethoprim is partly metabolized in the liver and will be excreted in the urine. Okay, what are the adverse effects of this drug? Nausea, vomiting, skin rashes can be appear due to this. Then this can be present or develop the folate deficiency that can result into the megaloblastic anemia. This effects we can prevent by the folinic acids. Okay, this is about the trimethoprim, which is the enzyme inhibitor. Okay, now in case of the sulfa drug, it is also the similar way mode of action like the trimethoprim. Okay, then the sulfonamide drug is the structural analog of the PABA. Only this much difference is there. Okay, in this picture, as I have mentioned, the red color one they have mentioned is sulfa drug, because the sulfonamide drugs are the analogs of PABA. Okay, this is the sulfonamide action. In the trimethoprim, what will happen? The pteridine, in place of the pteridine there will be the trimethoprim. Otherwise the reaction will be inhibited in the same manner. Okay, only the difference between both the drugs are the trimethoprim is a structural analog of the pteridine, whereas the sulfa drug is the structural analog of the PABA. In this online lecture we studied the enzyme inhibitor, trimethoprim, okay, and the sulfa drug. Now sulfonamide drugs are also broad spectrum bactericidal. The other properties are similar. Both the drugs have similar properties. Okay. They are also useful in the urinary tract infections that can be caused by the E. coli or the Proteus species, Proteus mirabilis. Okay. I hope you understood this online lecture. If you have any doubt regarding this, please ask in the comment section. Thank you for watching.